All right. Hi, Sham. So you're the only one here today. I'll just briefly go through what this course is for those who are joining or watching the video for the first time. So this is the week 6 problem solving session, and each week I take about 10 questions of questions that are from the module of that week. So in this case, it is questions that are from the week 6 module of the course Basics of Crop Breeding and Plant Biotechnology, and this is just to check the knowledge that you have gained or to understand how much of the topic you've understood and to solve any doubts that you might have regarding it. My name is Anakha Nair and I'm a PhD scholar at IITB, and I'm the one who will be taking these problem solving sessions each week. All right. So, without further ado, let's move on to the first question. And you can just try guessing the answer in the chat. I'll keep my chat open. So you can just try guessing the answer, and then we'll, anyways, be explaining the answer for each question. So no worries about that. So the first question is, mirin causes arrest in which phase of cell cycle? And you can stop me anytime you have a doubt or you don't understand anything, all right? So, like I was saying, mirin causes arrest in which phase of the cell cycle? And there are four options given to you: G1 phase, G2 phase, S phase, and M phase. So, what do you think the answer is? You can just write your guess in the chat, just to test yourself as to if you know the answer or not. That's all. All right, I'm not seeing an answer in the chat, so maybe I'll move on to the explanation. So, just in case you don't know what the different phases of a cell cycle is, so there are in total, you can say three or four phases, depending on if you count mitosis as a part of it or not, right? So the first part is the G1 phase. That is what you see here. And this is what happens to a cell immediately after it is formed. After a cell divides into two daughter cells, these daughter cells undergo the first phase, that is G1 phase. And that is where the cell growth happens, right? This is followed by the S phase. Okay, this is where the DNA synthesis happens, and that is where the DNA is, replicated so that when the cell further divides, it will have two copies of that DNA. Then this is followed by the G2 phase, where again further cell growth happens, and this is followed by mitosis, where the cell divides into two daughter cells, and again these two daughter cells go into the G1 phase, and that is the cycle that is followed, right? So the answer for this question is, Mirin causes arrest in which of the phases of the cell cycle? And the answer is G2 phase. Okay, so mirin causes arrest in this part of the cell cycle. Okay, and it does that by affecting something called or inhibiting the phosphorylation of something called ATM molecule, and this ATM molecule is responsible for the process by which double stranded breaks are repaired by the cell. So if the double stranded breaks that are formed are not repaired by the cell, then it gets stopped at this phase, G2 phase. Okay, since ATM is being inhibited by the mirin molecule, the double stranded breaks in the DNA. So double stranded break means if this is the DNA, a break in both the strands is what a double-stranded DNA is, and it uses a particular repair mechanism to repair this double-stranded break, and ATM molecule helps in that. But this mirin molecule is inhibiting this ATM molecule, so double-stranded breaks are not repaired. And if the double-stranded breaks are not repaired, the cell is getting stuck in the G2 cycle. Okay, so the cell arrest happens in the G2 phase. Was that clear? All right. So if that is clear, let's move on to the question number two. Just stop me anytime you have a doubt. All right. So question number two is, which of the following could be true in RNAi silencing construct? So, if you remember in a module, sir will tell you how RNAi silencing is used in the creation of the kind of what is it called? I'm forgetting the name. The RNAi is used to you know it's a step, right, in the reverse breeding mechanism. So if you remember from one of the modules, he had shown you how you construct the RNAi silencing construct. So do you have any clue to what the answer can be for this? So the question is, which of the following could be true in RNAi silencing construct? So which of these following is the construct that you will make and that you will use for the reverse breeding? So there are four options given to you. In option one, you have a promoter uh, followed by a gene in this direction, a linker again followed by a gene in the same direction, and then a terminator. In option 2, you have a promoter, you have a gene in the forward direction, you have a linker, again a gene in a forward direction, then the terminator. In option C, you have a promoter, you have a gene in the forward direction, you have a linker, you have a gene in the reverse direction, and the terminator. And in option D, you have a promoter, you have a gene in the forward direction, a linker, a gene in the reverse direction, and a UTR, which is the untranslated region. Okay, so one of these options is the correct one. So you have to choose one of them. So if you remember, this RNAi construct is supposed to form something called a hairpin loop structure, right? So that is a clue. Maybe that can help you figure out which of these is the correct answer. So let's maybe move on to the answer to this. So the answer is option C, where you have the RNAi silencing construct has a promoter region, a gene in the forward direction, a linker, a gene in the reverse direction, and a terminator. So what happens is this area will be translated into a RNA, right? Sorry, transcripted into an RNA, right? And since these two genes are in opposite directions, they will form complementary regions, right? And those complementary regions can bind to each other as double-stranded RNAs, right? And this linker region is what is forming this hairpin loop structure, right? So this is the construct that is going to work for this reverse breeding. That is the RNAi silencing construct, right? So if there are no doubts regarding this question, let's move on to question number 3. The question is, after transcription, the mRNA produced will be. There are four options given to you. Option A is similar to the coding DNA strand, where coding strand serves as a template. Option B is similar to the non-coding DNA strand, where non-coding strand serves as a template. Option C is similar to the coding DNA strand, where non-coding DNA strand serves as a template. And option D is similar to the non-coding DNA strand, where coding strand serves as a template. So for answering this question, there is a few things you need to know. What is a coding strand and what is a non-coding strand, right? So if this is your DNA molecule, one of them is the coding strand and one of them is the non-coding strand, right? So the non-coding strand here is the strand against which the mRNA will be formed. So imagine this is when the mRNA is being formed, right? And this is the coding strand and this is the non-coding strand. So complementary to this, the mRNA will be formed, right? Okay. And this is the non-coding strand or the template strand. The coding strand is this strand, that you see here, and it will have same sequence as the mRNA that is being formed, right? Except in DNA it will have T and instead in the RNA it will have U. Okay. Apart from that, the code will be the same. So out of these, option C is the correct one. That is, the mRNA produced will be similar to the coding strand, where the non-coding strand serves as a template. So like I was saying, if this is the coding strand and this is the non-coding strand, this will serve as a template, the non-coding strand, and mRNA will be formed complementary to this. And since this mRNA is complementary to the non-coding strand, it is also the same sequence as the coding strand, right? Because the coding strand is also complementary to the non-coding strand, right? So the mRNA and the coding strand will have the same sequence, except T will be replaced by U since it is a RNA and the coding strand was DNA, right? So C is the correct answer in this case. So if there are no doubts, let's move on to question number 4. Question number 4 is application of reverse breeding for isolation of chromosome substitution lines will be mostly practiced for the following conditions when there are four options given to you. Option A is reverse breeding is applied to heterozygous and heterogeneous populations. Option B is reverse breeding is applied to F1 hybrid of known parents. Option C is reverse breeding is applied to F1 hybrid of unknown parents. And D is reverse breeding is applied to F2 hybrids of known parents, right? So let's see what the correct answer is. The correct answer is B. Reverse breeding is applied to F1 hybrids of known parents. So in this case, you know the genotype of the parent 1 and the parent 2, and you have the F1 hybrid, and you use this F1 hybrid to perform the reverse breeding to get parents, a parental line that will give you um, upon crossing the same F1 hybrid, right? So the correct answer is B. Reverse breeding is applied to F1 hybrid of known parents, right? So let's move on to question number 5. So question number 5 is reverse breeding can be easily applied to, and the options are option A is crops with diploid chromosome number of 26. Okay. Option B is crops with haploid chromosome number of 13. Option C is crops with haploid chromosome number of 11. And option D is crops with diploid chromosome number of 42. So if you remember from one of the modules that Sir had taken, he had mentioned something like reverse breeding is easily applied to, and a certain chromosome number was mentioned, right? So let's see what the correct answer is. So the correct answer is crops with haploid number of 11. So haploid number, if you remember, is the chromosome number that is found in the gametes, right? So the chromosome number in the gametes is the haploid number, okay? And if you remember, Sir had said that anything anything having a haploid number of 12 and below, for that reverse breeding can be easily applied, and that is because it simplifies the isolation of the homozygous parental line, right? So when you're doing the reverse breeding, you need to find homozygous parental lines, right? And that is relatively easier to do when you have a haploid chromosome number of 12 or less. Anything more, it is very tedious for you to find the homozygous parental line. So if there are no doubts, let's move on to the next question. That is question number 6. So question number 6 is you have to choose the incorrect statement. So there are four options given to you, and you have to choose the incorrect statement from this. Option A is reverse breeding is required to maintain hybrid stability. Option B is reverse breeding involves selection of parental lines followed by production of desired hybrid plants. Option C is reverse breeding is required to maintain a hybrid's genetic makeup. And option D is reverse breeding is required to maintain the vigor of hybrid performance. Right? So you have to choose the incorrect statement. So let's see which the correct answer is. Right? So the correct answer in this case is reverse breeding involves the selection of parental lines followed by production of desired hybrid plants. Okay. So reverse breeding, like you can tell from the name, right, is where you have a desired plant, okay, and you have to create parental lines for that desired plant, right? So if you have created, so you have a F1 hybrid, okay, and it has the desired characteristics, so you reverse breed so that you get a parental line P1 and P2. Upon the crossing of these will again give rise to the F1 hybrid of the desired characteristics that you have. Okay, so it is not involving selection of parental lines, it involves the creation of parental lines. Okay, so option B is incorrect. Option A is correct. Reverse breeding is required to maintain the hybrid stability, which is correct. Option C, that is reverse breeding is required to maintain a hybrid's genetic makeup, is also correct. Okay. And option D is reverse breeding is required to maintain the vigor of the hybrid performance, is also correct. So if there are no doubts, let's move on to question number 7, which is the plants obtained by reverse breeding are cisgenic plants, genetically modified, non-genetically modified, and transgenic plants. Okay. So the correct answer for this, let's see what the correct answer is. The plants obtained by reverse breeding are non-genetically modified plants, right? So when you do chromosome doubling, okay, to induce reverse breeding, and it results in plants that are genetically identical to the parental line, right? And but you have used RNAi silencing to exclusively target any meiotic crossover. So the RNAi silencing that you are doing, it will stop, crossing over from taking place, like in this case. So no crossing over will take place, right? Okay. And when you're selecting from the gametes or from the, um, you know, um, from the gametes that you're getting, which to use to create the parental line, right? You are choosing one which still doesn't have the RNAi silencing gene, right? Okay. So in this case, they are not. So the plants that you obtain from the reverse breeding that you are choosing from the gametes formed, right, are not sorry, they are not genetically modified, right? So the correct answer is non-genetically modified plants. They are not transgenic or genetically modified or cisgenic plants, right? So they are non-genetically modified plants. So if there are no doubts, let's move on to question number 8, which is reverse breeding could be easily applied to which crop? There are four options given to you: cotton, lettuce, tomato, and onion. And the correct answer, let's see what that is. The correct answer is reverse breeding could be easily applied to onion. Okay. So the reason onion is the correct answer is because cotton, lettuce, and tomato are not. Okay. So this is because the double haploid plants are a very rare occurrence in these three plants. So it's very difficult to form double haploid plants in these. So it is not easy to perform reverse breeding because reverse breeding requires the formation of double haploids, right? So it is not applied to these plants. Reverse breeding cannot be done or cannot be done easily in cotton, lettuce, and tomato. Okay, and onion, since the double haploids can be formed for onions, we can use onions for reverse breeding. Like we can perform reverse breeding on onions, right? So let's move on to question number 9. Question number 9 is you have to choose the correct statement this time. There are four statements given to you. Reverse breeding accelerates the breeding process by starting with inferior hybrid selection. Reverse breeding method reduces available genetic combination and diversity. Option C is reverse breeding approach always involves screening and backcrossing plants having prior knowledge of their genetic constituents. And option D is reverse breeding process uses molecular markers for rapid trait detection. So only one of these statements is correct. So let's see which is the correct statement, right? So the correct statement is reverse breeding process uses molecular markers for rapid trait detection, right? So if you remember from the example, you will have a F1 hybrid and that will have certain molecular markers, and the gametes will have certain molecular markers, right? And from that you will choose, this is just an example, right? So I'm not doing it very correctly, but from that you will choose, maybe let me redo this. Hi Vishal, so maybe I'll take the questions from the top for you, right? So let's see what the first question is, and you can just write your guess in the chat box, right? So the first question is Marian causes arrest in which phase of the cell cycle? There are four options given to you: G1 phase, G2 phase, S phase, and M phase. So
M phase.
M phase, all right. So let's see. Let me just explain the different phases of the cell cycle, right? So the first phase is G1 phase, where the cell growth happens, right? Phase 2 is the S phase, where the DNA is doubled or DNA synthesis happens. This is followed by G2 phase, where further cell growth happens, and this is followed by mitosis, right, where the cell divides to give rise to two daughter cells. And the two daughter cells will start with the G1 phase again, and that is the cycle that is followed. So mirin causes arrest in G2 phase, that is here. So mirin is a molecule that causes the cell to not be able to repair double-stranded breaks. So double-stranded breaks in DNA are where both the strands are broken, right? So there is a particular way the cell is, repairing the double-stranded break in DNAs, and unless and until it can it can repair all the double-stranded breaks, it cannot move from the G2 phase to mitosis, right? So since mirin causes the cell not to be able to repair double-stranded breaks, the cell cycle gets stuck in the G2 phase. Okay, so the correct answer for this question is G2 phase. All right, any doubts?
No.
All right, sure. So let's move on to question number two. So the question is which of the following could be true in RNAi silencing construct? So if you remember from the module in the reverse breeding technique, you use RNAi silencing and you have a construct um, that is used for this RNAi silencing, right? So which of these four is the correct kind of construct that you need to create?
Two are the flanking regions, and middle is the linker.
Yes.
And at the start, there is a promoter. So, B option,
B or C? I didn't hear what you said.
uh, yeah, B, C option, I think.
Yes. So the
C
correct.
or B, I am confused.
Yeah, so I'll explain it to you. So the correct answer was in fact C, and like you said, it requires a promoter obviously and a terminator, so that the RNA polymerase can start and terminate the making of the RNA, right? And the reason C is correct is because it has a gene in the forward direction, there is a linker and a gene in a reverse direction. So what happens is this is the portion that will create the RNA, right? So
Hmm.
it will form something called a hairpin loop structure. Okay, since this is in the forward direction and this is in the reverse direction, right? They are
Right,
complementary to each other, right? So they'll form
right, right.
this, yeah. They'll form this hairpin loop structure. So you were correct. Option C is the correct answer. It's not option B because in this case it's both in the same direction. So this portion won't be complementary. This portion and this portion won't be complementary. So you won't form the hairpin loop structure. It'll just be a straight um, RNA molecule, right? So the correct answer was option C. So if there are no doubts, I'll move on to question number three. Okay. So after transcription, the mRNA produced will be, and there are four options given to you. Option A is similar to the coding DNA, where coding strand serves as a template. Option B is similar to the non-coding DNA, where non-coding strand serves as a template. Option C is similar to the coding DNA strand, where non-coding strand serves as a template. And option D is similar to the non-coding strand, where coding strand serves as a template. And they're asking what the mRNA will be like, right, after transcription. So transcription is when the DNA strand is used to make a mRNA strand, right? So, what do you think the correct answer will be? What is the mRNA that is produced will be like? Any guess?
A.
Okay, let's see what the answer is. So, before I move on, I'll just maybe explain what coding and non coding means, right? So, if this is the DNA strand, you have one coding strand and one non coding strand. The terminology might be confusing to you, right? So, what this means is it's the non coding strand where the mRNA is formed. Okay, so the mRNA is complementary to the non-coding strand, right? And since it is complementary to the non-coding strand, that means this mRNA and the coding strand have this, for this portion at least, the same sequence, right? Because the coding strand is also complementary to the non-coding strand. Since the coding strand and the mRNA are both complementary to the non-coding strand, it tells us that the coding strand and the mRNA are of the same sequence, right? Did you understand that a little bit? You want me to explain it?
Yes.
Huh.
No,
So,
no, yes.
huh. So that means the mRNA that is produced in this case, if this is the coding strand and this is the non-coding strand, and here the mRNA is being produced, right? So it is similar to the coding strand. That means it is the same sequence as the coding strand, and it is the non-coding strand that is serving as the template for the mRNA, right? So the correct answer is
Right.
option C. Okay. So if there are no doubts, let's move on to question number 4. Question number 4 is application of reverse breeding for isolation of chromosome substitution lines will be mostly practiced for the following condition when, and there are four options given to you. Reverse breeding is applied to heterozygous and heterogeneous population. Reverse breeding is applied to F1 hybrid of known parents. Reverse breeding is applied to F1 hybrid of unknown parents, and reverse breeding is applied to F2 hybrids of known parents. Do you want to try guessing?
Reverse breeding is applied upon hybrid of known parents.
Yes, so you are correct. It is correct. So reverse breeding is applied to F1 hybrid of known parents. Great. Alright. So let's move on to question number 5. Question number 5 is reverse breeding can be easily applied to, and there are four options given to you. Crops with diploid.
Chromo-
Huh?
Chromosome number less than 12. So
Yes.
crop with haploid chromosome number of 11.
Yes, so that is also correct. So chromosome number has to be of the haploid, that is the gametic chromosome number has to be less than 12. Only then can we easily find homologous parents, right? So in yeah, just one sec. Huh. So this is the reason why we need it to be less than 12 is because that will simplify your isolation of homozygous parental line, right? So you have to find parental lines that are homozygous, and it is easier for you to do so if there are 12 or less chromosomes in the haploid. If it is more, it will be very tedious for you to do it, right? So let's move on to question number 6. Question number 6 is you have to choose the incorrect statement, and there are four statements given to you. Reverse breeding is required to maintain hybrid stability. Reverse breeding involves selection of parental line followed by production of desired hybrid plants. Reverse breeding is required to maintain a hybrid's genetic makeup, and reverse breeding is required to maintain the vigor of the hybrid performance. So, one of these statements is incorrect. So, what do you think it is?
Means incorrect statement I have to identify.
Yeah, you have to identify incorrect statement.
Uh, 1 minute, madam.
Yeah.
Reverse breeding is required to maintain. Uh, Madam B.
Yes, so it is correct. Option B is correct. So option A, C, and D are obviously correct statements, right? So reverse breeding is required. We the reason we do it is to maintain the hybrid stability and obviously the vigor of the hybrid's performance, and it is also required to maintain the genetic nature of the hybrid. So you're making parental lines so that each time you cross that parental line, you get the same genetic makeup for the hybrid, right? So option B is the incorrect statement in this because it is saying that reverse breeding involves selection of parent line followed by production of desired hybrid plants, right? So that is not the correct statement, right?
Right.
Yeah. So that it is the ulta of it, right? So it is involves the selection of a hybrid plant followed by the production of parental lines, right? So you are correct in that case. So let's move on to question number 7. The plants obtained by reverse breeding are cisgenic plants, genetically modified, non-genetically modified, and transgenic plants. So what do you think the correct
Uh,
statement is?
Non-genetically modified.
Yes, you're correct about that also. They are non-genetically modified. Even though you're using RNA silencing method, the gametes that you end up choosing are those which don't have that RNA silencing done, right? So
Right.
essentially, the products that you get from reverse breeding are non-genetically modified. So yes, the correct answer is non-genetically modified. So let's move on to question number 8. Question number 8 is reverse breeding could be easily applied to which crop? Cotton, lettuce, tomato, and onion.
Madam, I don't have any.
Yeah. So this is slightly a memory based question because sir had mentioned which it cannot be applied to. So the correct answer is onion. So onions can easily form, double. Uh, wait. Um,
Double
yeah,
haploids.
correct. So the onion, there was one portion, if you remember, from sir saying ki certain plants like cotton, lettuce, and tomato, it is and soya bean also, right? So cotton, soya bean, lettuce, and tomato are difficult to form doubled haploids. So once you get the gametes, you're supposed to double the chromosomes, right? So it is difficult for that to be performed in plants like cotton, lettuce, and tomato. So the correct answer in this case will become onion. Okay. So let's move on to the next question. That is question number 9. So you have to you choose the correct statement in this case. Okay. And there are four options given to you. Option A is reverse breeding accelerates the breeding process by starting with inferior hybrid selection. Option B is reverse breeding method reduces available genetic combination and diversity. Option C is reverse breeding approach always involves screening and back crossing plants having prior knowledge of their genetic constituents. And option D is reverse breeding process uses molecular markers for rapid tree detection.
Madam B.
It's not B. So the correct answer in this case is reverse breeding process uses molecular markers for rapid trait detection. You remember that one portion where sir was saying if this is the genetic makeup of your F1 plant, okay, and you have gametes, right? And you see that you have gametes with these sort of traits, right? Okay, so you use this molecular markers to choose two plants, right? Maybe this and this. Okay, so you are choosing this as parent 1 and you are choosing this as parent 2, and you perform doubling of the chromosome, so that when you end up crossing this, you end up with this F1 hybrid, right? So the correct answer in this case is reverse breeding process uses molecular markers for rapid trait detection. Okay, so option B is not correct because it does not reduce available genetic combinations and diversity. That's not what it is doing. Okay, option A is also not correct because it is not. You don't start with the inferior hybrid. You start with the hybrid that has the most vigor, right? And option C is also not correct because it does not involve back crossing plants or having prior knowledge of their genetic constituents, right? So let's move on to question number 10. So the question number 10 is which of the following spore combinations will be used to produce the double haploid which upon recombination will yield similar F1 recombinants. So this is the F1 recombinant parent, right? And you performed tDNA insertion in line 1 and line 2, okay? And you end up with these gametes or these spores. So you have to find two such spores which you can use to form the same F1 hybrid that you had at the beginning. So,
No, no guess, madam.
no guess? Okay, so just in case the picture is not very clear to you, it's like this. So, the large chromosome that you're seeing, okay, it has this shaded portion and this shaded portion and the centromere in the center, right? The smaller chromosome that you're seeing is having shaded portion like this and shaded portion like this. Okay. So the correct answer for this will be first of all you need to remove anything that has the tDNA insertion. So tDNA insertion is shown by this yellow dot that you can see, right? So any plant that has the tDNA insertion, any spore that has it, you will reject, right? So these will be rejected because they had, you can see the yellow dot, right? That each of them had, apart from the centromere. So this portion, so just below the centromere, you can see this line, right? This line, it's visible, right? Like, are you able to see the picture clearly?
Yes.
Huh. So you will, since you don't want the tDNA insertion in the parental line, so you will reject these wherever you have the tDNA insertion in the spores. You will reject those. So you are left with one, two, three, and four options, right? So 1A, 1B, 2B, and 2D. So you have to find from them two such spores which upon recombination will give rise to this one itself, right? So let's look at option 1A. So option 1A has this portion of the chromosome that you can see, right? And it has this portion of the small chromosome. Okay. So it has a structure like this and a structure like this, right? So that is present in the F1 hybrid parent. And if you see option,
1A,
huh?
1A, 2D.
1A, 2D. Yes. So the correct answer is 1A, 2D, right? So if you cross 1A and 2D, you will end up with this recombinant, right?
Right.
Yeah. So that is the correct answer. So that is it for questions today. Do you have some doubts? Any doubts?
I have doubt in previous assignment. Let me check once minute.
Yeah, sure. Is the deadline for the assignment over?
I think.
Yeah, I'll just see because I'm not supposed to technically help you with the. This is from which week? I haven't seen the assignment questions for the current session, so I'll see if I'll be able to
No,
answer.
not current session, previous.
No, no, I'm saying for like these questions that I'm taking are, I haven't seen the assignment questions that you are actually getting for this. So I'm just
Okay, okay.
similar questions I'm taking. So let's see if I'll be able to help you. Which week is this from?
Okay. Let a minute.
Yeah,
I'm logging in.
sure.
Mm, Madam, in week number, assignment number 4, week 4,
Week 4. Okay, just let me see. Okay, the question is, which question do you have a doubt in?
question number 6, which crop is least likely to exhibit inbreeding depression?
Question number 6.
6.
Yeah. So which crop is least likely to
Least.
exhibit inbreeding depression?
So how we come to know in this that answer is wheat?
Yeah. So I don't know if you remember or not, but plants which
If the self-pollinated crops are there, then the inbreeding depression will be less.
it won't be there only because inbreeding
Yes.
depression means that if you keep crossing with yourself, you eventually end up with a very inferior line because all the recessive traits have come up, right? And these will be lethal and non-viable genes, right? So the reason why self-pollinated crops don't do that is because over the course of evolution they have come up with the way that they have the genes that they have, right? Even when they keep in like self,
But madam, here soybean is also self-pollinated crop, na?
is it?
I think two self-pollinated crops are given here.
Let me
Let me.
just check. Soya bean.
Soybean.
I am not also sure. So
Yes, it is self-pollinated, but they can also be cross-pollinated.
yeah, so I am now that you've correct wheat, soya bean. So yeah, technically soya bean can also be a correct answer since it is also a self-pollinating crop, right?
Yes.
So had you written soya bean as the answer?
No, I have written sugar beet,
Oh,
but
okay.
I was confused with that English word, the least likely
Ha ha ha. Understand.
or most likely. But concept, I am clear with the concept that if
Ha
it
ha.
is self-pollinated,
Pollinating,
then
then there's no
there are less
inbreeding depression.
chances of
Ha,
inbreeding.
correct. So it is wheat and soya bean technically. I don't know why wheat is the correct answer in this case. Because even in the answers, it's just mentioning wheat is a self-pollinated crop and naturally
Yes,
homozygous.
but soybean is also self-pollinated.
Yeah. So
If there is any technical reason, so they could be right there over soybean.
yeah.
How
So
wheat is the too much accurate answer.
so let me just check if they are homozygous in nature or not.
Okay.
Naturally, because the other thing that they're mentioning is that wheat is so they are technically not homozygous. So
Okay.
Google is telling me that soya beans, although grow as inbreeding, generally homozygous plant, the germplasm of the species contain large amount of genetic variation. So since
Okay.
they have a large, this is something that you might not know if it comes randomly in exam, obviously. So
Okay.
this is a slight trick question, I feel. But because wheat is naturally homozygous in nature, selfing does
Yes.
not cause inbreeding depression at all. A soya bean has slightly
Okay.
more chance because it has more genetic variation. So by inbreeding or by selfing, you might end up with some sort of inbreeding depression. So that is the
Okay.
only reason that I can think why wheat is chosen as because the question was least likely, right? So they're saying
Yes. Yes.
out of
Right.
the four, they're most least likely. Even though if there was the that they might not show inbreeding depression, wheat and soybean can both be the answers because they're self-pollinating. But since you had to choose the least one from these four options, I think wheat is the correct answer because it is homozygous in nature.
Thank you, madam.
All right. Any other doubts?
No, madam.
All right. Thank you so much for joining.
Thank you.
Yeah, you can leave the meeting now. Thank you.